of this realization, I decided to start working in science in order to get a better grasp of an understanding of the processes that lead to diseases. I became what's called an assistant scientist in Cosmetics at another major school. After finishing my medical studies at Göttingen, I moved to the United States as a postdoctoral fellow to work with two brilliant scientists, Mike Brown and Joe Gorski. Now at this stage in my career, I was not interested. I was interested in living. It's a very interesting part. I studied cholesterol metabolism, especially cardiology, biology, because the RDL receptor regulates the levels of cholesterol in the blood and the levels of cholesterol in turn determine whether or not you will get heart disease. I finished my postdoctoral studies in a couple of years and then became an assistant professor at UT Southwestern in Dallas, Texas. I also became an investigator of the Howard Hughes Medical Institute, which I still am. And in 2008, I moved to Stanford. When I initially moved to Dallas from Germany, it was unclear to many people why, especially in Germany, why I would want to move to Dallas, Texas, of all places. It is a pretty, let's say, unusual place for a European to go to. It is not blessed with natural beauty. What you see here is a picture of downtown Dallas in the background and Southwestern in the front. It's an outstanding place to do science because there's nothing else you can do. It's a great place to go to. This is what my lab looked like. I still look the same, as you can see. I haven't aged a bit. But after 25 years in Texas, I became tired of it, and I moved to Stanford as the Avram Goldstein Professor in the School of Medicine. Now, why Stanford? I think you can guess some of the reasons. Stanford is actually blessed with natural beauty. It is a wonderful place to live. What you can see here on the left is a picture of Stanford with the palm trees. But I think what's most noticeable about Stanford is the spirit of, I would say, freedom and discovery in academia. Despite the mercenariness of the surrounding Silicon Valley, despite the emphasis on money and profits and companies that is present there, Stanford itself has an academic culture that is very well enunciated in its motto, which turns out to be German, die Luft der Freiheit weht, which means basically the air of freedom blows. And this is really what attracted me to Stanford, the outstanding science that is being done there, and that I hope to profit from as I was moving my lab and my operation into that university. And that's where I've been for the last 10 plus years. So what have I actually been doing? I'm not going to tell you in detail any kind of special subjects. Instead, what I would like to do is give you a feeling for what I'm actually interested in my research and what the kinds of data, the kinds of results are that we have obtained. When Jim Rothman, Randy Shackman, and I were awarded the Nobel Prize in Physiology or Medicine in 2013, the citation said it was for discoveries of machinery regulating vesicle traffic, a major transport system of our cells. And you may wonder, listening to this citation, what has that got to do with the brain? How is that related to the brain? And actually it has a lot to do with the brain. It basically refers to the most fundamental process that allows nerve cells to communicate with each other in the brain. When you look at a brain, like this picture here of a dead human brain, what is immediately apparent is that the human brain is enormous in size, and it is composed of a huge number of neurons, each of which is connected to other neurons by thousands of synapses. This creates a complexity of networks that is unbelievably high, much more complex than, for example, the complexity of the human genome. As you all know, we do not understand the human genome at this point, despite its apparent simplicity. The idea that people had 20 years ago when the genome was first decoded, that we would now understand how genes actually work and how the human genome encodes a human, has turned out to be completely wrong. We have, in fact, very little ideas about how exactly the genome operates, and we know even less about how the brain really works. But there are some fundamental concepts that all scientists who work on the brain share. And the most fundamental of these concepts is the idea that the brain operates by processing information via networks, which are called neural circuits, and which are not completely different, although somewhat different, from circuits in a computer. What you see here schematically is what a circuit looks like. This is a microcircuit, just a very local circuit, showing blue excitatory neurons that are connected to red inhibitory interneurons. And what you can see in this very simplified circuit, in which the synapses connect these neurons into the network, is that even in a simple circuit like this, it is not easy to predict what the input-output relations of that circuit will be. Take, for example, this blue excitatory neuron that is shown on the left, and that has a recurrent excitation onto two different red inhibitory neurons. If you think about signals going through the blue circuit, the effect of this blue cell's excitation as a feedback mechanism via the interneurons cannot be predicted, because synapses differ. Synapses that connect these neurons into circuits have different properties. They can be facilitating or depressing. They can have all kinds of different properties whose precise nature actually determines the input-output relations of even a simple circuit like this. As a result, in order to actually understand how circuits operate, we need to understand synapses. And synapses are really what I'm interested in. All of my work focuses on synapses. What you see to the right of this image here is an electron micrograph of a single synapse to illustrate the point. Synapses are the basic computational units of the brain. They transfer information from one neuron to the next, and they also change that information. They process that information. They are like little microcomputers with a memory. Get the next slide here. Oh, oops. Okay, live grid feedback system. You don't see it on here, but it's on here. Do not allow, right? I'm having an IT problem here. Did somebody help? Okay, I think I got out of it. So, synapses differ in properties, but they all operate by the same principle. A principle that was first elucidated by Bernard Katz, who received a Nobel Prize for this discovery in 1970. And this principle operates as follows. When you have a synapse as shown here on the right, where you have one presynaptic neuron contact a postsynaptic neuron like this, what you see in the presynaptic side are lots of vesicles that are filled with a chemical messenger called a neurotransmitter. What happens is that an action potential, an electrical signal, invades the presynaptic nerve terminal on the left. It depolarizes it and causes the influx of calcium that then triggers the fusion of these vesicles with the presynaptic plasma membrane, releasing the neurotransmitters. The neurotransmitters then in turn react with postsynaptic receptors. This simple principle underlies all synaptic function. It is simple because it involves only the secretion of one chemical and the recognition of that chemical by another cell. However, it is less simple than it sounds because all of this has to happen within a few milliseconds and has to start very rapidly and to end very rapidly in order for neural circuits to operate. And this was the big question that we initially set out to solve. The question of how is it possible that a presynaptic neuron can do neurotransmitter release within a few hundred microseconds or milliseconds in a precise time-limited manner. Neurotransmitter release is mediated by the calcium-induced fusion of the vesicles with the presynaptic plasma membrane. This is the key step. And that is the step that is also plastically changed during computational processes at synapses. So how does that work? Let me explain this a little bit more detail in order for you to get a feeling for how we approach an understanding of the central process in information transfer at synapses. The first thing is that the vesicles need to actually fuse with each other at the presynaptic plasma membrane in order for neurotransmitter release to occur. The second element is that this needs to be regulated very tightly, quickly, on and off, by calcium. Very fast, 0.1 milliseconds, cooperative, you need multiple calcium ions. The third property is that it needs to be highly localized. The calcium needs to act only in one particular space and nowhere else. This is achieved by a macromolecular machine that organizes all of these processes together in a single space and enables this very rapid fusion process to mediate neurotransmitter release. Work over the last decades elucidated the principal machinery that does this. And it turns out that there's 3 separate nanomachines that together affect the 3 steps I just outlined. One machinery for the fusion of the vesicle, one machinery for the calcium regulation, and one machinery for bringing everything together and enabling it that everything would happen in a very localized space in a very short time. The discovery of this machinery that allows a nerve cell to mediate this process was really what was our key discovery that led to the Nobel Prize. Now, why would this be again important for understanding the brain? Why am I telling you this in a very general lecture? I'm telling you this because this machinery in the end underlies all communication between nerve cells. And that communication in turn mediates the workings of neural circuits. The brain is not only composed of one circuit or a few circuits in parallel. Millions of circuits that overlap or are interdigitated or in parallel, that are local or far distant. And all of these circuits are composed of separate thousands of synapses that have different properties. Understanding how these synapses mediate input-output relations of the circuits will require an understanding not only of the principles of the synapses operation, but also of their differences. Moreover, many brain disorders are thought to involve dysfunction of synapses and thereby change circuits and thereby cause the diseases, again necessitating an understanding of the underlying biology. Just to illustrate to you how complex this can be, what you see here is actually a simplified image. Looks pretty simple, doesn't it? Of a frontal cortex circuit in the brain. And so you can imagine that this complexity can only be understood if we understand the constituent synaptic connections, which is what we work on. So this was a brief summary of what my research is focused on. Initially on the fundamental processes that enable synapses to transfer information, more recently on a related subject, namely the question of how synapses actually initially form and how they change as a function of use and during disease. What I want to now come to is a discussion of brain disorders as it relates to our molecular understanding of the brain. And to introduce this, I just want to remind you that in all societies, disorders of the brain will dominate future medical practice simply because of the success we have had, and with we I don't mean really anybody related to me, I mean the world, has had in enabling people to live longer. There is more and more incidence, higher incidence in brain disorders, for most of which we have no therapies. In fact, for most of which we have no understanding of what is going on. What you see here is just a list of the disorders that I'm talking about, illustrating that they are coming in a large variety of different diseases. Neuropsychiatric diseases themselves alone account for the largest amount of years lost to disability, higher than any other diseases, higher than cancer. But they're very, very difficult to treat. Neurodegenerative disorders becoming incredibly prominent. And. Nearly all brain disorders are chronic and would benefit from treatments, obviously. Moreover, not only the age, but also the increasing complexity of our societies have led to an increasing prevalence. For example, brain disorders are a large increasing problem, as shown here in the slide that was picked from an article in The Lancet 10 years ago, illustrating that even then, and since then even more so, there has been an enormous burden for society from neuropsychiatric diseases. These diseases often do not have advocates in societies because they are thought to be not that bad. They are thought to be not as threatening as cancer, for example. But let me tell you, these diseases are just as devastating for people as cancer. I want to illustrate this for example, in two images from Vincent van Gogh's work 150 years ago. Here, illustrating a man with depression, which I think very beautifully, if that can be called beautiful, shows what an enormous sadness this disease elicits, what an enormous suffering. Another example, a self-portrait of Van Gogh with his ear cut off, or half cut off, illustrating an episode he himself experienced that probably represented schizophrenia, of tremendous suffering. And as you know, Van Gogh committed suicide because he couldn't, in the end, bear this suffering of his disease. Possibly more important, the neuropsychiatric disorders for all societies, owing to our increasingly aging population, are neurodegenerative disorders. These are becoming more and more prevalent. What you see here, again, is taken from an article from 10 years ago, just to illustrate how huge that impact will be in 10 years. Even India is on there, and you can see the impact is predicted to be enormous. And this is just for Parkinson's disease. With Alzheimer's disease, it's even worse. So the fundamental challenges here is to understand the disorders in order to develop strategies for preventing them, therapies for stopping progression, and possibly avenues for regenerating brain function. There is no major treatment available for most diseases. In fact, I would like to argue there is no understanding of these diseases, which makes it very hard to develop treatments. Let me again use a couple of examples to illustrate the challenges. As you all know, Alzheimer's disease is a major threat. As you grow older, the incidence in Alzheimer's disease rises exponentially beyond the age of 80. And at this point, despite billions of dollars, we have no real treatment. What you see here is an overview of the projected economic impact of Alzheimer's disease. I'm not going to go into the details here. Let me just tell you, it's going to be severe. What happens in the disease is that nerve cells die. In fact, it is thought that what happens first is that synapses die. And because the synapses die, the nerve cells die afterwards. This process leads to a shrinking of the brain, a loss first of memory, and later on of all brain functions until the patient basically becomes completely incapacitated. It is a devastating disease that leads to a very rapid loss of any individuality of the patients. The brain shrinks because nerve cells die, but we don't know why the nerve cells die. When you look at it in a microscope, what you see as illustrated here is that in Alzheimer's disease, the nerve cells do not look normal in that they have material inside them that look like tangles and are called neurofibrillatory tangles. In addition, in the extracellular space, there are plaques, which are called amyloid plaques. Plaques are largely composed of a peptide, a protein component called A-beta, that may be toxic for neurons. The discovery that A-beta was present in these plaques led to a large investment of research into A-beta, into an enormous attempt in clinical trials to target A-beta as a way to treat Alzheimer's disease. The idea here was, as shown here on top, that Alzheimer's proteins are toxic, in this case A-beta, because they form aggregates, which are visualized as these plaques. and that if you add an antibody against these toxic A-beta aggregates, that you can sort of detoxify these aggregates. As a result, the major current treatment ideas aimed to decrease the toxic amyloid using antibodies. But all trials have failed, despite costs of exceeding billions of dollars. Moreover, even if an antibody therapy for Alzheimer's disease was available, it would be hard to imagine that a wealthy society like the United States would be able to afford it, let alone less wealthy countries where the problem is just as severe. The reason for these failures is simply that all of these trials were based on the hope that A-beta is a toxic component that kills neurons, and that battling A-beta would actually help the disease. But there was no real evidence that this was the case. In fact, there was no real understanding that Alzheimer's disease was actually due to A-beta and that the neurons died because of A-beta. In other words, all this work on translation, on developing a drug, was done without an understanding of the disease. And it's an illustration to me that it's pointless of trying to go to translation if you don't really have an understanding of the underlying biology. So you can invest a lot of money in some possible treatments that may lead to nothing because you don't really understand what's going on in a patient as in Alzheimer's disease. Let me show you a different example, a positive example that illustrates the same point. When you have an understanding of the underlying biology, you can actually attack a disease. This disease is multiple sclerosis, which is, although it happens in the brain, in fact, not really a brain disease. It's an immunological disease. And we can treat it because we have a good understanding of immunology. Multiple sclerosis is caused by a focal inflammation that destroys the myelin sheath that surrounds axons, which in turn mediate the communication from a cell body of a neuron to its presynaptic terminal. This immunological process, autoimmune process, destroys these axons secondarily and thereby causes enormous brain dysfunction. This is not uniform in the brain, but happens in little foci, as illustrated here in this MRI picture, where you can see with the arrows foci of inflammation where a patient with multiple sclerosis experiences localized inflammation causing the disease. It is a chronic autoimmune disease for which excellent drug treatments are now available because we know how to treat the immune system. It's a multi-billion-dollar market that has led to a large number of drug options, which I'm not going to go into in detail here, but which are listed here, that are both small molecules and immunologics, and that have been able to treat a large percentage of the patients, although even with this successful example, there are many patients who are resistant, and there's still need for further drugs for this disease. So the lesson that I want to communicate here is that brain disorders are a huge challenge and a major opportunity for therapies. They are a vast market, obviously, because so many people are affected. But for most diseases, we have no understanding, and thus there is no rational way to design therapies. There's really nothing to translate. We need the biology first. Even for diseases for which there is a broad understanding, sort of, like multiple sclerosis, epilepsy, the available treatments are only partly effective. And so the bottom line here is that much progress was made in cancer, that whereas much progress was made in cancer, there are only small advances at this point in treating brain disorders. And that's primarily because the science is not there, because we don't understand what's going on, because there hasn't been much investment in it. So let me come to the last part of my talks today, in which I want to tell you a little bit about how I think we as a community in biomedical research need to approach this. And before I go into what I believe, let me explain to you how I see the drug discovery process based on my work both in science and in industry. I think that the relation of science to innovation is not direct, it's indirect. It is indirect because science is problem-oriented, it answers a question, it doesn't want to solve a practical solution. Science is not directed towards solving a practical solution, it basically wants to generate knowledge. Engineering, on the other hand, is completely different. In engineering, you know what solution, what practical problem you want to solve. You're product-oriented. You make a product using science applications once you have the science. This distinction is important because you need good science to get a product, but you can't get good science by focusing on a product. You can't start off with, I want to solve this, and then do the science for it. And I want to illustrate this with an example that comes from my own experience when I trained as a postdoctoral fellow. In the 1970s, it was known that increased cholesterol leads to heart disease because it causes atherosclerosis. And so it was clear that here's a problem looking for a solution. The problem was, how do you develop a drug that lowers cholesterol? And indeed, 20 years later, statins were developed. These are extremely effective and by now very cheap drugs that have saved millions of lives because they have prevented heart disease, as you see here. The logical approach for an engineer would be to engineer drugs that lower cholesterol. But that is not how it worked, and it is not how it worked because there was no idea of how to lower cholesterol. You can't just develop a drug against cholesterol. That doesn't work. You have to actually understand cholesterol. The way how it worked was that Brown and Goldstein, in whose lab I trained, addressed this problem by first studying cholesterol metabolism and then devising ways based on those scientific results to engineer a solution, to actually develop drugs that lower cholesterol levels. In other words, the way this worked was that they first identified an important question: how is cholesterol regulated? Then they did the research over decades without a specific hypothesis, trying to figure out how cholesterol is regulated. And this led to the discovery of one particular enzyme as the key regulatory event, HMG coenzyme A reductase. This was turned over to the drug company who developed a drug based on the science that would inhibit HMG coenzyme reductase, the statins, and then clinical trials confirmed that these drugs were actually effective in treating hypercholesterolemia. So what you see here is a process where the actual discoveries could not be planned or engineered, but were essential for subsequent engineering. In other words, you need the science first before you can move on to translation. Scientific progress cannot be scripted. You have to basically let scientists ask their questions and pursue them. With this drug discovery principle in mind, where are we going in the pharmaceutical industry and how that relates to neuroscience? In the remaining few minutes of my talk here, I want to give you a personal perspective of how I think this will proceed. I want to tell you about what I think are the current trends in CNS drug development and where this will go in future. And I think that the biggest challenge is to do the science first that identifies a target that is druggable and relatively disease-specific. And that is really where we have to start in and where we have to invest the money. Now, recent technological advances have enabled us to make enormous progress here, because there have been new techniques in neuroscience that have really changed our perspectives, techniques that developed over the last decade. Stem cell biology has enabled studying human neurons that was previously not possible. Genomics has enabled looking at genetics of humans for diseases that was unprecedented before the sequencing costs came way down. Now we have large-scale sequencing for almost everything, enabling us to follow large populations with different diseases and leading to an enormous treasure trove of information that are sometimes used good and sometimes are used commercially. Transcriptomics has changed the way how we look at single cells. We now can actually look at single-cell RNA-seq data that allows us to define what genes are being used in any given cell at any given time. On the other extreme, structural biology, cryo-EM, has really changed how we actually look at molecules, how we can see molecules, how we can define their atomic structures. Antibodies, the most time-honored, oldest tool for any biomedical research, discovered more than 100 years ago, have also been revolutionized by the development of multivalent antibodies and of nanobodies that promise completely new classes of biologics. All of these tools depend on basic biology first. Applications will emerge later. Moreover, the types of drugs that are being developed are changing rapidly. As you all know, for a long time, small molecules have dominated medical practice. In fact, in India, I believe, they are one of the major industries in biomedical field. And they still supply most of the useful drugs. However, in most R&D and most pharmaceutical research and most biotech, small molecules are no longer being pursued. The advantages of small molecules is that they're cheap to produce, that they can be brain penetrant, they're easy to copy. They have, in many ways, all the properties that you need of a good drug. And for example, it is no accident that statins have become such a great drug because they're cheap, because they're a small molecule. The disadvantage of small molecules is that they have many side effects, invariably, unpredictable often. It is very expensive to develop small molecules. It is very difficult to develop them. They have unpredictable PK and PD. They're just incredibly difficult to deal with, which is the reason that the large pharmaceutical companies shifted their emphasis on biologics, which are basically proteins like insulins, but primarily antibodies. These are now the focus of most biopharmaceutical research and development. They have predictable side effects, predictable PK and PD, and are easy to develop. They're cheaper to develop. But they're difficult to manufacture and to copy and standardize. And they're very expensive, making it almost impossible to use them for chronic diseases that happen to a large segment of the population simply because they wouldn't be affordable for any country. What we are now experiencing, however, is a revolution in that there's the third class of molecules, which are RNA and DNA therapeutics that are now being developed. And I don't think they're sort of shifted down at the bottom of the slide in this particular computer, and that are really changing the types of drugs that are the future. I think that we are now shifting increasingly over the next 2 decades towards developing drugs that are based either on some kind of cell therapy or, more importantly, on RNA or DNA therapeutics. For example, I believe that the most promising treatments for neurodegenerative diseases will probably be antisense oligonucleotides or related drugs that allow suppression of specific expression of molecules such as, for example, A-beta, instead of using antibodies. This is really where I think the futures and the opportunities lie in drug development for especially neuroscience applications, because biologics will never really work in this space. And I believe that this will revolutionize the industry. Drug companies service society obviously and importantly by developing and selling drugs. And obviously drug companies want to make money. But the way drug companies work in any given country is shaped by market forces, by rules and regulations, by the law, which can make the relationship between drug companies and society difficult or easy. Issues here are that because of high margins and long exclusivity, new drug development is focused on biologics at this point for cancer or rare diseases. This has tremendous benefits, but given the fact that cancer, despite its threat to the individual, is really just one of many diseases that our societies suffer from, is too limited. We need other types of approaches, other types of molecules. We need regulations that favor, for example, treatments for diseases of the brain. We need regulations that enable gene therapies. We need regulations that make it easier to develop these, that make it more profitable to develop these. Moreover, in the US at least, the healthcare delivery system is inefficient and unequal. We need better access to drugs that are based on preventive medicine. We need better models of regulating the dispensation of drugs for a society. So there's much to do for our aging societies that I think our and the next generations will have to address to combat the current increases in the diseases of the brain. So to summarize this part of my presentation and to come to an end here, which I'm sure you will be happy to hear, deep biology is an absolute prerequisite for development of any therapies. There will be no translation of neuroscience if there is no basic neuroscience. We need to understand the disease in order to identify targets that we can use for treatments. There are now huge new opportunities in identifying drug targets, new technologies that enable studying these diseases and the underlying biology, new types of drugs, especially RNA, DNA drugs, that we all should make use of. But we need regulatory mechanisms that ensure that drugs are really developed to the diseases that are important for the society as a whole. And I would argue that there should be much more emphasis on diseases such as Alzheimer's disease or schizophrenia, which have such a large burden of suffering but so little treatment options. And with this, I would like to close. I've tried to tell you a little bit about my own research and my own background, and I've tried to tell you a little bit about where I think neuroscience needs to go in order to help society dealing with pressing medical issues. And I hope I've convinced you that the most important goal should be, if we want to get good translation of science, we need to have science first. Thank you very much.
Thank you very much. This way, please.
Thank you, Professor Sudhaf, for your excellent talk. Professor Sudhaf narrated his research of last 30 years in a lucid manner. His work initially focused on the synaptic transmission and whose molecular basis was completely unknown at that time. Later on, Sudhaf's work increasingly turned to the analysis of synapse transmission and specification, processes that mediate the initial assembly of synapses, regulate their maintenance and elimination, and determine their properties. In this process, he identified a number of key molecules in pre- and post-synaptic neurons. In his last part of talk, he talked about the problem of drug design. He rightly pointed out that science answers the question. Science is not directed to solve classical problems. Another word he rightly mentioned, that discoveries cannot be planned or programmed. For the students, I would like to tell that no scientist start his work to get with something in mind that he will be having Nobel Prize. If you go through the Nobel Prize winning discoveries, you will find that a major part of the Nobel Prize discoveries were chance discoveries. They were working on some problems and they got interesting results in the process. With these few words, I would like to thank Professor Thomas again for his excellent talk. Thank you, Professor Thomas.
Thank you, Professor Chakravarti, and I personally understand that he has very nicely explained the complexity of the brain. Do you remember that circuit diagram which he has shown? And very nicely, this is the power of being a Nobel laureate, you know. You explain the most complex phenomena in the most simplest form of it. Give a good round of applause for Professor Sudhaf. I take this opportunity to request Professor Abraham Hershko, another Nobel laureate with us. Give a good round of applause for Professor Abraham Hershko. I would like to request Dr. Manoj Kumar Chakravarti to join him onto the stage, along with Professor K.S. Rangappa, the former Vice Chancellor of Mysore University and President-elect Indian Science Congress Association. I welcome you to this second session of Nobel Laureates. I request Dr. Ramesh Thakur, quiet please. Please take your seats. I need not to tell you once again this. Come back to your seats. request Dr. Ramesh Thakur to extend a floral welcome to our Nobel laureate, Professor Abraham Hershko. Dr. Ramesh Thakur. Professor Ramesh Thakur to kindly welcome the General President, Dr. Manoj Chakrabarti, by offering him a floral welcome. I request Dr. Ramesh Thakur to kindly welcome Professor K.S. Rangappa, the former Vice Chancellor of Mysore University and the General President-elect, the Indian Science Congress Association. Thank you, Dr. Thakur, for the warm welcome. Let me hand over the podium now to the chair, Dr. Manoj Kumar Chakrabarti, the General President, Indian Science Congress Association, for further proceedings. Over to you, Dr. Chakrabarti.
I understand this is also the first visit of Professor Hershko to India, like Professor Sudhaf. And here, I should introduce Professor Rangappa. He was already, Professor Loviraj Gupta has mentioned that Professor Rangappa is our General President-elect. He will be the General President for the next 2019 to 2020. So he will organize the Indian Science Congress Association next in 2020. Professor Rangappa is an well-known scientist, and he has been working for the last several years on the identification of small molecules from the plant extracts. And recently, some of you might be knowing that two of his contribution to the results, and their results has been published in PNS. He identified two small molecules which can be a candidate drug against cancer in future. With these few words, I would request Professor Rangappa to introduce Professor Abraham Hershko.
Good evening, everyone. Dignitaries on the dais, and off the dais, ladies and gentlemen, media friends. It's my proud privilege as a chemist to introduce the Nobel laureate, Professor Abraham Hershko. The Nobel Prize in Chemistry 2004. Professor Abraham, born on December 31st, 1937, in Hungary. His affiliation at the time of the award with Technion Israel Institute of Technology, Haifa, Israel. His main discovery on the discovery of ubiquitin-mediated protein degradation. The Nobel Prize shared by three of them. The main discovery, an important process in our cells, is the production of proteins, but proteins must also be broken down. At the beginning of the 1980s, Abraham Hershko, Aaron Schicknover, and Irwin Rose showed that one protein, ubiquitin, has a special mission in this context. When it is sometime for a protein to be broken down, a ubiquitin molecule attaches itself to the protein. The ubiquitin molecule serves as a key that enters a proteasome, a protein complex that divides the protein into smaller pieces. These can be used in the construction of other substances in the cell. Now, I request Professor Abraham Hashko to deliver his talk. Thank you, one and all. Thank you.
That is not my slide. Anyhow, good afternoon, everybody. I am very, very happy to be here at the beautiful campus of the lovely professional university. I'm also very happy to be in India, a beautiful country, a rich culture from which we can learn a lot, all humanities, I think. So I would like to tell you briefly about my work, and then I would like to discuss with you what can we learn from this work. So I started out, like Dr. Sudhoff, I started out by studying medicine. I wanted to be a medical doctor. But during my studies as a medical student, I, as all medical students, had to learn a lot of medical sciences, and I got interested in it. I saw there is so much still to learn about medical problems, about the major problems in medicine, cardiovascular diseases, cancers, that have to be solved by research. So I went on to research. Still, I can compare medicine to what it was when I studied. That's not mine. Can I have my first slide, please? That was the previous one. Yeah, that's it. Okay. Yeah, that's the first slide. And then I start right here. Okay, so when I compare medicine as it was 50 years ago to what it is now, there is some huge improvements. There was a large increase in life expectancy, which increased by about 10 years, so which is a huge increase in this time. And this large increase in life expectancy was to many improvements. There was a great improvement in the prevention of diseases. For example, in cardiovascular diseases, the use of statins that we just heard about it decreased cholesterol levels, which produced a lot of cardiovascular diseases. The management of high blood pressure, hypertension, is much better now than it was before. And most importantly, our lifestyle is much better than it was. Now we know to avoid high-calorie diets, high food and high fat diets. We know that smoking is not good. All these are very important in our health. And then there was also great improvements in the diagnosis and early detection of diseases, which are so important in treating cancer. And that is mostly due to improvements in technology. For example, when I was a medical student, there was no CT, no computerized tomography. There was no MRI. There was no ultrasound. All these are very important technological improvements which are important for early diagnosis. Also, molecular diagnosis is much better now than it was before. Now we can have genetic tests which can prevent all kinds of genetic diseases. And of course, there was a great improvement in the treatment of disease. Many of these is due to technological related again, like the intensive care units that decreased mortality a lot, pacemakers, for example, and all kinds of novel drugs, such as now we have targeted cancer therapy, we have immunotherapy for cancer, which are great improvements. We can treat AIDS with protease inhibitors, which will convert this fulminant disease into a chronic disease which can be managed. So there were great changes and great improvements in healthcare in the past 50 years. And all these are due to a lot of research and development. And when we look at the role of scientific discovery in the improvement of healthcare, we can distinguish between basic research and applied research. Now, basic research provides a foundation for great advances in medicine. For example, there is the example of the discovery of penicillin by Fleming. It was during his basic research on bacteria, when his bacterial plates got infected by a fungus, and he noticed that around the fungus there is a killing of bacteria, and that was the base for the discovery of penicillin, which was produced by the fungus and killing bacteria. So that was the first antibiotic, and which came many other antibiotics. Another example in how basic research helped later on applied research is the discovery of MRI by Lauterberg, and the story there is that he was a young chemist, and when he had some dinner or something in a cheap restaurant, he looked at the glass half of water, and that gave him the idea that by MRI, by nuclear magnetic resonance imaging, he can tell the state of the water in the glass, and that was the beginning of the discovery of MRI. So basic research is always the basis for applied research, and here I agree with the previous speaker. And then we have applied research, which benefits directly healthcare and other things in science. For example, in medical sciences, applied research is coronary stents, laparoscopic and robotic surgery, and new antibiotics, and all that is based on discoveries in basic research. So all the improvements in medical science is due to basic research, and then the basic research is the basic for the applications. And as an example of how basic research or fundamental research can give rise to application, to improvements in medical care, I will show you my own work. I understand this is a mixed audience. Many of you, of course, very intelligent, but do not have background in biomedical sciences. So I try to be I will try to avoid jargon or as much as possible, and to explain to you my work, and then we'll try to find out, I try to discuss with you what can we learn from this work. So my work, as you heard, is about protein degradation, and first I will try to explain what are proteins. Proteins are the so-called machines in our body, and they carry out all the processes, all the different processes in our body. All the different processes are chemical processes, and there are thousands of different chemical processes in our body which are all essential for life, and these are carried out by proteins called enzymes. So we have thousands of different enzymes that carry out thousands of different chemical processes. Proteins also carry out all the physical processes, like the contraction of our muscles, like, as you heard, the working of our brain, different electrical activities that are all carried out by proteins. And proteins are the regulators there are the regulators of, for example, cell division or of the embryonic development. And of course, the immune response, all the antibodies, we have thousands of different antibodies that fight against thousands of different invaders, and all these are proteins. So we have many thousands of different proteins in our cells, all of which have different functions. Now, how can we have so many proteins? For that, we have to know how proteins are built. And proteins are built of building blocks called amino acids. And here I showed only 4 of these building blocks. There are 20 of these. And in the protein, these amino acids are linked together like beads on a string. And the length of protein is several hundreds of amino acid building blocks, and the difference between the proteins is the order or the sequence in which the different building blocks are arranged on that string. And the sequence is determined by our DNA, by our genes. So the genes are the blueprints that give the instruction to the formation of the different proteins. And this process of assembly of amino acid building blocks to build a protein is called protein synthesis. And when I started to work in science, there was a lot of interest in protein synthesis, in how the proteins are made out of the different building blocks. So there were many people who were interested in protein synthesis. But there is an opposite process in which the proteins are broken down to their building blocks, to the amino acids, and this is called the protein degradation. So when I started out, there was a lot of interest in protein synthesis, but very few people were interested in protein degradation. But I saw that protein degradation is also important, and it is important because of 2 main processes. That doesn't, okay. 2 main processes. In one important function of protein degradation is to remove abnormal proteins. So proteins, after they are made, they are folded into a specific structure, and this specific structure is very important to their function. But this structure of the proteins is very delicate. It can be very easily damaged, and then the protein becomes abnormal. And when it becomes abnormal, it is toxic to our cells. It can kill our cells. So it's very important to remove abnormal proteins. And one important function of protein degradation is to remove abnormal proteins. But if there is another important function, is to degrade completely normal proteins that have to act for a certain period of time. So it's like switching off a function. Yes, there is a protein that has to work only for a certain period of time, but then it has to be removed. It's like our cell is like a big orchestra in which the different proteins, there are thousands of different proteins, and they all have to work together, all have to work in symphony. And when they do not do that, then a certain protein works only for a certain time, and then it has to be removed. Okay, so my question was, I see that the audience is beginning to get a little tired, so I think I'll go over this. So this is ubiquitin, and the discovery was that this protein marks which protein has to be removed at a certain time by being linked to it. And that is, okay. Okay. I didn't show it. Okay.
This one?
No?
Number, this one? See it on here, sir. This slide. This slide you want to see?
Yes.
This
The slide that was here was not there?
Yeah, here, sir.
Ah, okay.
Here, sir.
So there was, okay, so the problem was that I was still talking about different slides which were not shown. So that shows the importance of technology. Okay, but, okay, I'll go a little bit further. So the question was how to degrade a certain protein at a certain time, and the discovery was that there is one protein called ubiquitin that marks which protein has to be degraded. And that is the shape of the ubiquitin molecule, which is here. Here it has the, on the upper right side. Okay. So this shows you how ubiquitin works. Ubiquitin gets linked to the protein that has to be degraded. And once it is linked to it, there is a large structure called the proteasome that degrades only proteins that are linked to ubiquitin. That is the main thing. So that was a discovery in basic science. And then I think we are running out of time. Then it was important in many basic processes, such as cell division. I skipped that. So then many people got interested in this system, and it was found out that ubiquitin-mediated protein degradation is important in many important processes in which a certain protein which regulates has to be degraded at a certain time. And these are, for example, the control of cell division when cells have to divide, that is determined by the degradation of certain proteins. Then cells have to get signals transduced from outside to the inside of the cell, and that is also carried out by the ubiquitin system. The ubiquitin system is involved in almost every important cellular function. And so when I started out, not many people were interested in this process, but then many people got interested in it because it turned out that almost every basic cellular function is carried out by the ubiquitin system. And then the medical community also got interested in it, and it turned out the ubiquitin system is involved in many major diseases, such as in many types of cancer, because ubiquitin system is involved in the control of cell division. It's also involved in cancer, and many types of cancer are due to abnormalities in the ubiquitin system for protein degradation. There are many major neurodegenerative diseases, such as Parkinson's disease or Alzheimer's disease, are due to the accumulation of abnormal proteins in certain parts of our brain. And when they usually these abnormal proteins are removed very fast, but in these cases they are not removed quite well, and it is so that the accumulation of these proteins has at least a part of a role in these neurodegenerative diseases. Then ubiquitin system is involved in some types of mental retardation, some viral diseases, and also in muscle wasting, or in the wasting of muscles, which is called cachexia, which happens in many bad diseases. So because the ubiquitin system is involved in many major diseases, the pharmaceutical companies or industry became interested in it, and now there's already a drug that is based on this basic discovery. The drug is which is efficient against a certain type of cancer. The drug is called Velcade or bortezomib, and it inhibits the proteasome, which is a part of the ubiquitin system, and it was found to be highly efficient in the treatment of multiple myeloma, which is a bone marrow cancer. And this is a cancer that in our bone marrow, the cells became cancerous, and they kill the function of the bone marrow to produce blood cells. They also damage the bones, and it is an awful disease that used to kill people or the patients within a year or two. And now, because of this drug, these people can live many more years of good quality life. So the drug, the Velcade, does not cure them, but converts an acute disease, a bad disease, to a disease that can be controlled for many years, and because of that, it became a drug of choice for the treatment of multiple myeloma. So, as you see, after many, many years, my work and the work of my colleagues helped, in which we had a basic process. We wanted to know how proteins are degraded and how is the selectivity of protein degradation is carried out. But because we found out the system, then it became a target for drug discovery, and that is an illustration. It also gives me a lot of personal satisfaction that my basic research now can help many thousands of sick people. Of course, that is only the first example, and now the drug industry is very busy to try to find other targets which are based on this basic discovery of the ubiquitin-mediated protein degradation. So that is my story, and now what can be learned from my story? What are the lessons from this story, mainly for the young people in this audience? So one of the lessons that I learned, I think, that it is very important to have good mentors in science. Yeah, you cannot learn how to do good science just from reading the literature. Of course, you have to learn the literature. You have to know what was done, what was not done, what is the basis for what you want to know. But to learn to science, you have to learn it from persons. You have to know how to approach a problem, how to do good experiments to find out some answers to your question. And for that, you need good mentors. So I was very lucky, very fortunate to have good mentors in my early life in science. That is the Professor Mager, Jacob Mager. He was my first mentor. He was my mentor for my PhD thesis in the Hebrew University in Jerusalem. And he was an excellent biochemist. He was a biochemist of incredible knowledge. He knew almost everything about what should be known about medical sciences. He was also a biochemist, a very fine and rigorous biochemist. He taught me, and as his other students, how to do an experiment, how to plan an experiment with all the positive controls, the negative controls, so that you should be sure that your result is really so. And then each result had to be confirmed from all angles. So I am very grateful to Professor Mager for giving me a firm background of rigorous biochemistry. And then I went on to do my postdoctoral work with Gordon Tompkins at the University of California in San Francisco, where I first got involved in protein degradation, and I got him, and he was a very different mentor. He did not care so much about controls, and because of that, he got into trouble many times. But he had a lot of imagination, a lot of inspiration, a lot of getting out of the box. For every problem he had some brilliant suggestions what can be the answer. So I think that in science it is very important to have creativity, to have originality. But it is also very important then to study, to test with a lot of self-criticism whether your idea is indeed correct. So I was very, very fortunate to have these two very different mentors in my early life. Mager with a strong, rigorous biochemistry, and Tompkins with his brilliance. Together, I think I got a good mixture for these two types of approach to science. Now, once you had your mentors and you finished your studies, your PhD, and also postdoctoral studies, I see that it's studies, you have to become an independent investigator. And for the first thing is to find an important subject. And to find a subject for research, I think it is very important, very important to find a subject that is not yet in the mainstream. Find an important subject that is not yet interesting to others. Otherwise, the big guys will get there before you. Now here they got much before me. We have some problems in technology here. No, back. No, back. Oh, okay. So I am now at number 2. I won't touch now anything here. I am now at number 2. Find an important subject that is not yet interesting to others. Otherwise, the big guys will get before you, and do not go with the mainstream. So as you remember, when I started to do science at the Gordon Tompkins lab, everybody was interested in protein synthesis. It was a big lab. There were 25 different postdocs who were working on the synthesis of one protein, one certain protein from different angles, but on the same problem. And I saw that these too congested, and I wanted to do something else. And then that is how I began to study the degradation of that protein. Do not do what everybody else is doing. Try to do something that is different, but you should be convinced that this problem is indeed an important problem. Now I go to number 3. That accidental observations are the most important ones. I jumped over it because I saw that I am too late. So the story is that when I was there in my postdoctoral work, I studied the degradation of this protein, and by accident I found out that the degradation requires energy. And it was quite unexpected for me. I thought actually that I have some opposite result, but I found out that energy was needed for the degradation of this protein and for protein degradation in general in cells. Now that was surprising because protein degradation outside of the cells that was known, like the digestion, did not require any energy. So that's why I thought that maybe we have here a novel mechanism that requires energy for the degradation of proteins in cells, and that was actually the beginning of all the work on the ubiquitin system. The energy dependence was the first indication that we have here a completely new system. So that was an accidental observation, but sometimes an accidental observation is the most important one, and it is the one that really led me to the discovery of the ubiquitin system. And then you should need, number 4, you should need whatever experimental approach is needed for your objective. Now in science, as in many other things, there are fashions, there are modes, there are things that are now fashionable, technologies that are now fashionable. Now, you should not go after the fashionable technology. You should go after the technology that you really need. So that is my next lesson from my life in science. And then, number 5, science should be driven by curiosity. When you go into science, you can see there is a lot of drudgery. You have to write a lot of things. You have to write grant applications to get a grant. You have to write papers in order to get promoted. You have to collect a lot of data in order to write a paper. But you should not do only drudgery. You have to do all that in order to survive, but you should not do only that. And to do, I think the main thing in science is to follow your curiosity. It should be a lot of fun. And if you do things to follow your curiosity and not just for the drudgery, then you may make a great discovery. And my last lesson is never to leave benchwork. Now, benchwork is doing experiments by your own hands, and many senior scientists do not do any more experiments by their own hands. They let this to the younger people to do it. I think that is not very good, because you lose touch with how to do things. And you actually, when you do the same experiment by your own hands, you have much closer connection to the problem. You want to find out the result of your problem. You are still excited by your science. And for example, I never left the bench work. I'm still doing, whenever I can, I'm doing an experiment every day with my own hands, and that gives me a lot of satisfaction. But of course, you cannot do everything by your own hands. And here I would like to acknowledge some people who helped me for a long time. And in my lab in the Technion in Haifa in Israel, all these people helped me for many years, and including my wife, Judy Hershko, who is here, who worked with me for many, many years in the lab. And I had many graduate students who have worked with me, more than 35, among them Aaron Chehanover, who was my graduate student during the times of the discovery of the ubiquitin system of the ligation of ubiquitin to proteins, and because of that, he shared the Nobel Prize with me, so he is a graduate student. So if you do a PhD student and you are lucky, you may get a Nobel Prize. And of course, I had a lot of collaboration and help, among whom Erwin Rose was my colleague during the discovery of the ubiquitin system, and he shared the Nobel Prize with me. And now I have, in my present lab, I have these people who still help me and still work a lot about the roles of the ubiquitin system in the control of cell division. Thank you very much.
Thank you, Professor Abraham Hershko, for his excellent, popular talk on a very complicated subject. We know that, we understand that he identified a protein, ubiquitin, that marks degradation of other proteins. For students, I am telling that how a drug is developed. Initially, he received Nobel Prize in 2004. Okay.
Students, just wait for a while. Now this proceeding is only for 10 more minutes. I'm sorry to interrupt in between, but take your seats, please, for a while. It's just going to go long for 10 more minutes.
It was identified, discovered in 2004, before 2004, because in 2004 he received Nobel Prize on this discovery. And after that, you see the involvement of this protein in signal transduction, regulation of gene expression, and it was involved to inflammation, etc. And it also was involved in protein quality control by removing undesirable other proteins. It is also involved in the diseases like cancer, neurodegenerative diseases, viral diseases, muscle wasting, etc. And it took several years because lots of scientists were involved in these studies. And ultimately, he has shown that a drug has been developed, Velcade, which acts on multiple myeloma. And ultimately, that was a very good thing that he shared his experience and he gave some lessons to the students. With this, I would like to thank Professor Abraham Hartsko again for his illuminating popular talk. Thank you.
Thank you, Dr. Chakrabarty. We are at the verge of completion of this event. But I learned a very wonderful word from Professor Abraham, that is, don't be after fashionable science. Because science can be very, very fashionable. So you need to stick to your basic sciences. I take this opportunity to call upon VC sir because we are launching an international journal. I request VC sir to launch that journal right here. May I request Chancellor and Pro-Chancellor ma'am also to join VC sir while we are launching this international journal of ours. And Nobel laureates, kindly join the proceedings of the launch. And the chairs, kindly join. We are launching the international journal.
Good evening. First of all, let me congratulate the entire team who is standing at the podium, Lovely Professional University and ISCA, for a very, very successful beginning of the Congress. Congratulations. And it has happened because of the efforts of all of you. This is a wonderful occasion that we are talking about science, and Lovely Professional University would like to initiate its first international journal of recent development in sciences, which will be launched today, because ultimately whatever science we create, it has to be made usable for others by publishing papers in the journals. Thank you very much. And there's an international advisory board, but one of our faculty members, Dr. Praveen Kumar Sharma, from the Department of Chemistry, he will be the editor-in-chief of this journal. Praveen? And Praveen has been working very hard from last 2 days, and we didn't want to start the process without talking to Professor Chakrabarty that is it possible? And he said it's a perfectly good time to start this journal. Thank you.
Thank you, sir. Thank you so very much. May I request all other dignitaries to please take seats so that we can start. We are now commencing the proceedings of felicitation of the session. I request Honorable Chancellor LPU Pro Chancellor LPU and Vice Chancellor LPU to kindly felicitate the Nobel laureates first. I request Professor Abraham Hershko to move on for the felicitation ceremony. Please. I would request Mrs. Abraham to also join, because that's a gracious moment for all of us, that you have been here and you are enlightening the audience. And for the medal as well, there's an Indian Science Congress Association medal, which is given every year to the Nobel laureates. Give a good round of applause, please. What a moment that you. What a great, grand moment. You've seen Nobel laureates. Thank you, Professor Abraham and Mrs. Abraham. It has been great and kind. Thank you so much. May I request Professor Thomas Sudhoff and Dr. Chang to join us for the felicitation ceremony. Please do come. Please do come, Dr. Chang. And you know, we'll request Dr. Chang to interact with you during her sojourn at LPU, because she is again a professor at Stanford University. Dr. Sudhoff. So two brilliant minds together. Give a good round of applause. And the medal from the Indian Science Congress Association is being conferred upon Professor Sudhoff. What a great move. What a great move. Thank you, Professor. May I now request Dr. Manoj Kumar Chakrabarti. Now I'll be facilitating. I'll be asking you to facilitate all three of you. I request the Honorable Chancellor, worthy Pro-Chancellor and respected Vice-Chancellor, sir, to felicitate Dr. Manoj Kumar Chakrabarty, the General President, Indian Science Congress Association. Yeah, Mrs. Chakrabarty. No problem, you can have another. In the meantime, may I request Mrs. Saxena to please be on stage. Now I request Honorable Chancellor, worthy Pro-Chancellor and respected Vice-Chancellor to felicitate Professor Ashok K. Saxena and Mrs. Saxena, the past President, Indian Science Congress Association. May I now request the Chancellor, the worthy Pro-Chancellor ma'am and the respected Vice-Chancellor sir to felicitate Professor K.S. Rangappa, former Vice-Chancellor, Mysore University and President-elect, Indian Science Congress Association. Thank you so much, dignitaries. Thank you so much, Nobel laureates, and thank you so much, audience. You need to move out from the front gates so that we can have another audience coming from the reverse gate.